Welcome to our first post-pandemic forum. We, we thank our sponsors, the League of Women Voters, the American Association of University Women, the Nevada Appeal, a volunteer from Carson High School, and the Brewery Arts Center. Just a couple of quick housekeeping things. Please turn off your phones. And the restrooms are located in the lobby to the left. And the Nevada Appeal is recording this so that if you have friends and family who still want to see what it is and couldn't come, it will be on our website eventually, and maybe on theirs too. Our speakers are experts who are peer-reviewed, and some of our topics are controversial, but we try to present the facts. We have cards for questions, and he's holding the box of cards and pencils. So if you have questions, please write them down. We screen them here at this table for so that we don't have duplicate questions. And we expect to have 4 more forums between now and next summer. In October, we're going to do one about the Carson City School curriculum. In January, we're doing one about senior living options in Carson City. In March, we're going to talk about the Nevada Poverty Safety Net. In May, we're going to talk about how the infrastructure money is or will be spent in Nevada. And then in October, we'll have our regular candidate forums. So that's kind of our lineup. And now I would like to turn over our program to one of our beloved local physicians, Dr. Tim McFerrin.
Thank you for that warm welcome. Last time I got that much applause when I announced I was retiring. Well, we're privileged tonight to have some excellent physicians here who are very much renowned in their field, and experts in their field. And we're going to be talking about heart, eventually I hope lungs and cancer, but I know there's an absence of female parts up here, so we're going to start off with Kristen Gurney, who has a Master of Public Health from University of Michigan and is currently the Manager of Clinical Research Programs at Reno Health.
Okay, I'll just hold on to this guy. Okay. I don't think I've got my slides yet, but it's so nice to meet all of you, and thank you for being here today. So I'm going to talk a little bit about the centralized research office that we have at Reno Health and the affiliation we have with UNR Med that supports the provision of clinical trial opportunities in our community. So I've been with the research office for about 4 years now, and the affiliation was signed about 3 years ago. So it's given us a big opportunity to integrate medical education and clinical research opportunities into the various clinics that we work with. So I'll just go a little bit off script. It's totally fine. So our goal really is to provide a clinical trial for every disease indication that we see in our community, and making sure that the community is really central to the kinds of trials that we're bringing on in the office. Our primary focus right now is in oncology, cardiology, pediatrics, pulmonology, and neurology, but we're really open to working in any area. We also have an infectious disease study or two, so it's really diverse and really based on what the patients need in our community. We are working to facilitate medical student and resident research in our office as well. So not only are we working to support the community, but also to help grow that next generation of clinician researchers and continue to get advanced care within our community and also globally. I have a team of about 20 research coordinators across the affiliation and our administrative staff that help support that function so that it makes it easy for patients to participate and for researchers to bring on that novel care for our community and really advance what we are able to provide to the community, not just as far as standard of care, but give you an option to participate in something new as well if you're interested in that. So we can jump a couple of slides. That's my lovely team right there. So we're all here to support you in the community in Northern Nevada. We can go to the next slide. So at our UNR Med office, that office is focused primarily on investigator-initiated studies, which are local studies. We have a provider or a faculty member that has an idea, and they want to see if that's successful. That's what we foster there at the university, as well as the student and resident partnerships. Next slide. And then at Renown, through Renown Regional and at Renown South Meadows, we have about 111 clinical trials right now available to patients. And so, like I said, those studies are in 5 primary specialties with a couple of stragglers, and we're continuing to grow that specialty focus as well. We just onboarded an endocrinology study we're really excited about. So we're looking to expand out to every need that there might be in the community. Next slide. We do incorporate our research into the clinic setting, so it makes it easy for providers and patients to participate, just alongside any of your standard care that you would normally receive. Next slide. And like I said, we are working to educate those clinicians that are in the clinics currently, like current providers, as well as those upcoming providers, the medical students and residents in the research process as well. Next slide. And when we think about clinical research, I want to note that it's not just novel treatments or drugs like we would consider that you're getting from the pharmacy. It can also be related to the way that care is delivered in the inpatient setting. So it's really diverse. It's not always going to be investigational medications, but most of our studies are. But it can also be observational studies to see if something is working in the care setting now that it's gone to market, or if there are new ways to detect cancer, for instance, is one of our bigger studies. So it's a great way to find new novel markers that'll help us detect that early. Next slide. And then a big key part of my job as well, and that's why I'm here today, is to help provide that education to the community that we do have this care available to you, and talk about what's of interest to you so that we can be able to support the community the best way that we can. And next slide. I do want to make a note, too, that diversity in research is really important to us, and it's really important to the clinical trials industry as a whole. So we are developing a diversity statement for our office in alignment with the FDA's guidance to do so, but because it's the right thing to do, fundamentally, that's what we'd like to do moving forward. And so that's why we're here to collaborate with you as well and learn what you're interested in so that we can build that diversity statement together. My contact information is up there for both sides of our office if you have any questions, and I'm also happy to take questions later on during the Q&A session. So thank you.
Thank you, Kristen. And just one, if someone in the audience was interested in a clinical trial, this is where they would contact you?
Absolutely.
Okay, great. So our next speaker is Dr. Michael Block, cardiologist by trade. And at last, I think cardiac disease is still the #1 killer in the U.S., so it'd be interesting to see what's happening with cardiac research.
Yes, despite my best efforts.
Yeah.
So actually I'm a vascular medicine physician, which is related to cardiology, the better part of cardiology, actually. So one thing I will ask, I have a date tonight with my lovely wife of 26 years, so I'm going to be breaking out of here a little early. So if you have questions specifically for me during this talk, if you wouldn't mind just jotting them down, and if it's okay with you, I'll take them right after this presentation. So I'm going to tell you about a couple of clinical trials that we're doing at Renown. They're actually part of international clinical trial programs. It is super important that we support clinical trials. If you are taking a cardiovascular medicine, I guess with the exception of aspirin and digoxin, any cardiovascular medicine that you're taking is now available to you because it was in a clinical trial. And our clinical trials have become better and better designed. They've become more diverse in terms of the patients we put them in. They've become a little bit more refined in terms of the outcomes that we look at. But the concept has been the same for decades. We don't want to be using medications unless we can document in a really clear way that they are safe and efficacious. So I want to tell you about a couple of things that we're studying at Renown, if you can throw my slides up there. One in the field of, as a vascular medicine provider, I'm very interested in hypertension and in cholesterol, and so those are the two of the things we're going to talk about this evening. Next slide, please. You can go ahead to the next slide. So this is a slide that looks at the failing grade that providers like myself get in the management of high blood pressure in this country. We have had great medications. We know the lifestyle management that's needed to reduce high blood pressure. We know how important high blood pressure is. Uncontrolled high blood pressure leads to increased risk of stroke and heart failure and cardiovascular death. And yet we do a terrible job of treating high blood pressure in this country. As you can see, 43% of people with high blood pressure are controlled in this country today. That's a failing grade even in the Washoe County Public School District. We really do a poor job. And for decades, literally for decades, I started my career about the beginning of this slide, I've been going around and talking to patients and talking to providers about we really just need to take more medicine. More medicine, more medicine, more medicine. And quite frankly, I think our blood pressure medications are great, and I spend most of my time prescribing them, but it's very clear that that strategy is not working very well. The data is right here. So we need to expand our toolbox in terms of how we treat high blood pressure. Next slide. So one of the things that we're very interested, there are a lot of reasons why blood pressure is not well controlled. Certainly part of it is that patients don't like to take medications. Oftentimes, this is not the one medicine they're taking. They may be taking 2 or 3 medicines for their blood pressure. They have to take those every day. They usually have other illnesses as well. So the laundry list of medications could get quite long. And quite frankly, us as providers are often reluctant to increase the number of medications we're asking our patients to take. So for that reason, if you go to the next slide, we're very interested in looking at interventions, durable, one-time interventions to lower blood pressure, rather than always need to add another pill, add another pill, add another pill. There are a couple of different strategies that are out there. We're studying one currently at Reno, which is ultrasound-based renal denervation. So it's this cool little catheter that's right there. I'll tell you how that gets inside the artery in a minute. But basically that catheter that's there, that balloon on the end of that catheter, goes into the kidney artery, and ultrasound, that little copper area in the middle of that, is an energy source for ultrasound radiation. Ultrasound radiation is really hot, so it will burn whatever is around it. The renal nerves run along the renal artery, so our idea is that we are knocking out those renal nerves, which we know carry traffic, sympathetic nervous system traffic, to the kidney to raise blood pressure. But of course, we don't want the artery itself to fry. We want the tissue around it to get hot, but the artery to stay cool, cool as a cucumber. So what we have here is a water-filled balloon that constantly circulates cold water while we're doing the procedure. So the next slide. What we do is we can create this circumferential energy, this tube of energy, out about 1 millimeter to 5 millimeters outside the kidney artery. So what we do is we take the catheter, we do an approach where we go in through the groin, like if you had a coronary catheterization or get a coronary stent placed or something like that. It's the similar procedure where we go up to the renal arteries, we squirt some dye in so we know where we're going. We put this catheter into each side, we circulate that cool water, and then 2 or 3 times in each renal artery for 7 seconds, we have that ultrasound radiation that goes out and ablates those nerves that travel to the kidney artery. Next slide, please. So we have done a number of these studies. This is the results of the 3 worldwide clinical studies that we've done, the main results of those studies. We see that we can lower blood pressure. This is 24-hour ambulatory blood pressure. It's actually daytime ambulatory blood pressure, which is a specific way of measuring blood pressure. It's about 6 to 8 millimeters reduction by this methodology. If you look at traditional clinic blood pressures, which are always a little bit more exaggerated, we're looking at about 10 millimeters of blood pressure reduction, and very consistent in all the studies that we've done. Next slide, please. And super importantly, the safety and tolerability of this procedure appears to be bulletproof. We really haven't seen any major side effects or any major safety issues. That doesn't mean that we know it's going to be safe, right? That's why we continue to study it, and patients who are in these studies are followed for 5 years to make sure that they don't have any long-term safety issues. But to date, and we're out 3 years, 4 years in many of these patients, we haven't seen any major safety issues. Next slide, please. So this has been quite a journey. I'm actually on the International Steering Committee for this set of studies, and I work as a consultant with a company that makes this denervation catheter. I've been working with them for about 13 years. We've been doing these studies for a long time. And we've just recently submitted our results to the FDA. The panel voted that this looked like it was safe and effective, and they've recommended that the FDA approve it for routine clinical use. And so it's going to be another, weapon in our toolbox against the fight of high blood pressure. It's a little bit unclear, though, how it's going to fit into our current delivery system in terms of reimbursement, in terms of out-of-pocket expense, and things like that. That's something that's going to have to be worked out. But in the meantime, next slide, because we've been such a good site for this clinical trial program, we want to continue to gather data, mostly on safety, but also on effectiveness, while the FDA is deliberating, while Medicare and commercial insurance companies are trying to figure out how and when they're going to cover it. So we are involved in what's called a continued access program, where we are able to take and for essentially no out-of-pocket cost to our patients, patients who are eligible, have poorly controlled high blood pressure, top number above 140, bottom number above 90 on the medicines that they're taking. And if they want to take part in this clinical trial, which means being followed for 5 years to make sure that this is efficacious and safe, we're able to continue to offer this therapy at Reno. So this continued access program is really important, and it's going to get us even more data between now and when this comes to market. Next slide. So changing gears completely, let's talk a little bit about what's going on in the field of cholesterol management. I want to talk a little bit about the Ocean A clinical trial that we're a part of at Reno. Next slide. This is a trial that is looking at lowering an unusual cholesterol molecule. This molecule is called lipoprotein A. It's probably the most important molecule, cholesterol molecule, that you've never heard of. Lipoprotein A is really coming to the forefront as a major undiagnosed risk factor in lots of patients who end up having cardiovascular disease. Next slide. So it's, in many ways, if you look at it under a microscope, it is similar to LDL cholesterol, which is the bad cholesterol, but with a couple of important differences. It's got this funky apolipoprotein A that's wrapped around it that may make it not only lead to developing atherosclerotic plaque, that is the blockages that lead to heart attack and stroke, but also may make platelets a little bit more sticky. And if you have any atherosclerosis, any buildup of cholesterol plaque, it may make you more likely to form a blood clot on that, have a heart attack, and have a stroke. Importantly, it's 100% genetically determined, runs in families. Diet doesn't affect it. Exercise doesn't affect it. It is not picked up on a traditional lipid panel. And currently, we have no therapies that really lower it significantly. Next slide. So one of the things I think, getting a lipoprotein A measured is important. If you have cardiovascular disease in your family, if you yourself have cardiovascular disease, the Europeans say that this is such an important molecule that everyone should have their lipoprotein A measured once in their lifetime. That's what's in our current European guidelines. US guidelines haven't quite gotten that far this yet, but you heard it from me tonight. Eventually they're going to get there. It's inexpensive and it gets you a lot of valuable information, about 35 bucks, and usually covered by insurance. So once again, one of the reasons that we haven't traditionally measured is we haven't had a great way of affecting what are we going to do for patients who have high lipoprotein A. Traditionally, what we've done is we've kind of doubled down on our current lipid-lowering medications. We've tried to drive the LDL cholesterol even lower, so we'll use higher doses of statins and other medications to get the LDL cholesterol down. But we certainly would like to have drugs that could lower lipoprotein A, especially in these patients where it really seems to be the only risk factor that they have, and in patients with super, super high levels. Next slide. So this has been a target of evolution of our drugs for some time. And this current drug that we're studying is a small interfering RNA. really sort of a sniper shot to try to decrease the levels of lipoprotein A in our patients. So it's a small snippet of RNA that is given in a way that it goes straight to the liver. The liver is what makes your cholesterol molecules. So that little snippet of interfering RNA goes right to the liver, and it binds, if you remember your biology, when you want to make a protein, right, the cell will make messenger RNA to cause that protein to get made. What this small interfering RNA does is it gets into those liver cells and it binds up the messenger RNA specifically for lipoprotein A, not for anything else, just for lipoprotein A, so that liver cell stops making lipoprotein A. So this is the drug that we're looking at. There's a couple of other strategies that are out there. This is the drug that we're looking at. It's made by Amgen. Next slide. So we know that this drug, that given every 3 months in an injection, lowers lipoprotein A by at least 90% in the dosage that we're studying. We know that. We've already proven that. Lowers lipoprotein A by 90%, and it's well tolerated. A couple of injection site reactions sometimes. Super, super well tolerated. But that's not enough. We want to know that not only does it lower lipoprotein A, we want to be sure that it also prevents heart attack and stroke. Next slide. So that's the purpose of this study that we're doing. It's called the OCEAN-A endpoint study. It's taking patients who have a history of heart disease, specifically a history of a heart attack or a blockage in the coronary arteries that required stenting, and taking those patients who also have a high lipoprotein A, and we're looking at people who have really high lipoprotein A levels, twice upper limit of normal. We're taking those patients and we're randomizing them to either getting this medication or getting a matching placebo, and then following them for 3 to 5 years to prove that this molecule not only lowers lipoprotein A, but that it lowers heart attack and stroke risk. So this is a little bit further out on the horizon in terms of when it may become available in routine clinical practice, but also I think has a great potential to save lives and prevent cardiovascular events in our community. Next slide. One last thing I will say about lipoprotein A, it's not just about coronary disease. It's also not just about my generation of blocks. I have felt, and others have suggested for many years, that lipoprotein A is important not just in coronary disease, but we also know that it increases the risk of valve problems. And we knew it for aortic valves, but I was always under the impression that it did for other valves as well. And there's many of us who believe it increases the risk of blood clots like DVT and PE. So one of the things we were able to do, this is my son Evan at the American Heart Association, one of the things we were able to do at Renown is we were able to harness the power of the Renown electronic medical record and some of the data crunchers that we have at Desert Research Institute, and we were able to do a study of our patients at Renown who had high lipoprotein A. And what we were able to show is that, yes, or my son was able to show, he's the better doctor in the family already. Actually, I'm the third best doctor in the family. There's my son, there's my wife who's a neurologist, and then there's me. Better than the cats, though. Much better than the cats. We were able to show that high lipoprotein A levels in our system are associated not just with coronary disease, but with all valvular heart disease and with blood clots as well. So this drug that we're developing may have other potential targets, not just patients with coronary disease. So thank you. That's really all I wanted to share with you. Next slide. Happy, like I said, I got to run in a minute, but if it's with the moderator's permission, I'll take a couple of questions.
Well, I'm surprised.
Before I run.
Well, I'm surprised you didn't bring your wife here for a date. There's wine and chips out there. I mean,
This is probably as romantic as our date at the Killers tonight, but yes, we're going to do that instead.
First question is, please clarify the incidence of renal artery stenosis.
So renal artery stenosis is a relatively uncommon cause of high blood pressure. What renal artery stenosis is blockages in the arteries to the kidneys that are caused by atherosclerosis, the same thing that leads to coronary disease, the same thing that leads to stroke. It's a small percentage of patients with hypertension who have it. Interestingly enough, currently it's an exclusion criteria for use of renal denervation, because we don't really know what's going to happen when you apply that ultrasound energy to a patient who has a significant blockage. So that's one of the things we look for before we do renal denervation, is to make sure that there's no blockages there. So it's sort of a different topic of conversation, but I have spent a lot of my career studying renal artery disease. It was my first publications were in renal artery disease back in New York in the 90s. Those of you who remember the 90s.
This next question is kind of a technical question. There must be a electrical engineer out there, or some ultrasound engineer. This is regarding ultrasound renal device. What's the operating frequency of the device? What's the input power feeding the device? What's the reachable temperature in the immediate vicinity of the device?
All right, so I got to run. I am not an electrical engineer. I'm a history major. So I don't know the answer. I have known. I have them in my slides. I can actually, if you want to leave your email address, I can get you that technical information. It heats up to, it's for 7 seconds, but it does heat up quite a lot. Not quite to the boiling point, but it gets pretty darn hot in that narrow strip for about 4 seconds. It's super, super hot for 4 seconds. Sorry, I don't have all those technical answers.
And this is a question about the lipoprotein A and LDL. Can people have a high lipoprotein A and a normal LDL, or do they both have to have both a high lipoprotein A and a high LDL?
Yes, that's a great question. So we find that most patients who end up having problems because of high lipoprotein A also have a high LDL cholesterol. But you don't have to have a high LDL cholesterol to have problems with lipoprotein A. Particularly things like stroke in the young, we'll see in folks who have a high lipoprotein A with a normal LDL level. So they do appear that they are genetically linked through some genes, but in some patients they will be independent, if that makes any sense. So we kind of think about them independently. Does that make any sense at all? Very
Makes
little.
sense, yeah.
Very little.
So the answer is yes and no.
Yes and no. And don't forget maybe.
Yeah. So I have a question about using the renal ultrasound device. How successful were you getting patients off their hypertensive meds?
It's a great question. The idea behind this device, as you see, it's a relatively modest effect. It's also an intervention, right? It's not something that we take lightly doing in patients. It's going to be somewhat expensive. It's not the most fun day that you'll have in your life if you're undergoing renal denervation. So the idea really isn't that we're trying to get people off of medications. The idea is that we're not going to have to increase their medicines. So the idea would be that we would fit this in a patient who's on medicines but whose blood pressure is not yet controlled. And rather than increasing their medicines, we would offer them renal denervation to avoid increasing their medications. There have been some instances where we've been able to stop people's medicines, but that's not the expectation that I'm trying to set with our patients, either as part of this research or when we subsequently use it in clinical practice.
A couple more questions.
Do we have time for that? Okay.
Yeah. Would you recommend everyone getting the lipoprotein A test, like Europe does?
So like I say, I try to be a good soldier. I've worked with the American Society of Hypertension, the National Lipid Association, American Heart Association. I try to promote their guidelines, but I think we're behind Europe in this. I do think everyone should get their lipoprotein A level checked. I really do. Once, all you gotta do is get it checked once. It's genetically determined, so if it's high, it's high. If it's low, it's low, and it's gonna be that way the rest of your life. So I do recommend getting it done.
Is the new drug for hypertension using this similar messenger RNA technology that created the COVID vaccine?
The lipid drug?
Yes.
No, it is different technology.
Looked pretty similar.
Well, so many things in biology use RNA and DNA. This is a small interfering RNA. It's really very different than the COVID vaccine. And it's actually not really exposed to the immune. It's kind of the opposite. You're trying to hide it from the immune system so it doesn't break it up. So it's kind of the opposite of what we're trying to do with the COVID vaccine, which is show material to the immune system. So it's very different, actually.
So are there any other questions from the audience? Well, thank you, Dr. Block. That was very interesting.
I want to thank you guys for coming. Thank you very much.
And I hope you have a wonderful evening tonight. Great date. Thank you so much. Appreciate you taking your time.
Cool.
Thanks a lot. So our next speaker is Dr. Lee Schwartzberg. He's the Section Chief of Medical Oncology Administration at Renown Health, and he's been brought to Renown to expand the oncology unit there.
Good evening, everyone. First of all, I have to say I'm super impressed with the quality of those questions. I didn't know we were going to have electrical engineers, vascular biologists, and sociologists here. So I'm going to take a little different path to the tenor of my talk, and I'm really going to talk in a broader sense. And so you've seen about particular trials that Dr. Block showed you, which are really interesting. I'm going to give you kind of more of an overview of how we do trials, and particularly as it relates to cancer. So next slide, please. So this is the current cancer incidence and death rate for this year in the United States. And we're going to have 2 million new cases of cancer and 600,000 deaths. So we're number 2 to cardiovascular, but cancer, like it wants to be number 1, we're trying to avoid that as much as we can here. And you see the numbers for both males and females. The cancers differ a little bit. I will point out, though, that lung cancer is the number 1 incidence cancer for both men and women. And it's something to keep in mind. Of course, breast cancer, because there are number 1 cause of death for both men and women. Breast cancer is the highest incidence in women, and prostate in men. Next, please. So the good news is that cancer death rate is decreasing. Since the early 90s, the death rate has decreased by over a third. The number of people getting cancer is going up because as you age is actually the most important risk factor for cancer. And baby boomers, like myself, are hitting prime cancer ages now in the 70s, late 60s, 70s, and early 80s. So we expect to see, it will not be a crisis, we're expecting it and we're hoping to manage it as best we can, that over the next 10 to 20 years, we'll see more cases of cancer. But hopefully, based on clinical research, we will reduce the death rate more and more. And cancer will be cured more. For patients with advanced cancer, they will have their disease controlled more. We now talk about most cancers that are metastatic as chronic diseases to manage, sometimes in much the same way as you heard about managing hypertension. We use medications or sometimes devices to manage them as well. So there are currently 5 pillars of cancer treatment. Some of them are very old that have been used for decades or even centuries, like surgery. We've had about 100 years of use of radiation treatment, dating back to Madame Curie. Cytotoxic chemotherapy came into the fore after World War II, and the recognition that certain chemicals that were used could kill cancer cells. The molecularly targeted therapy started in the 90s. I'm going to focus a little on that. And then most recently, we have immunotherapies, which is listed here as the 1990s to present, although truthfully, the first experiments and the first results with immunotherapy date back 100 years as well. We just didn't understand the immune system well enough to create drugs that reliably would help us there. So how do we do clinical trials? And how does a drug even make it to a clinical trial? It starts with this bench to bedside practice paradigm. So generally, basic researchers will discover something, and they'll create a hypothesis about that discovery. And then they may develop a drug that relates to that discovery. So you find a gene that causes cancer, and then you look for a drug that inhibits that. A lot of work is done in the preclinical space. That means any studies that are done in test tubes, on cells, in petri dishes, in mice, in other animals. And then it gets to the bedside, which is clinical research. And we have to go through a whole process, which I'll get to in a minute, about how we do clinical trials in people. And then it gets to clinical practice. But it's not a one-way street at all. As you can see, when you're in practice, you learn more because only a small number of people have actually been exposed, relatively speaking, to the drugs until it becomes approved by the FDA. And then many more people get it under less restricted conditions, people with other situations, other comorbidities. And we learn more from that. And then it goes back in an iterative cycle back to the bedside. So we talk about bench to bedside. We also talk about bedside back to bench. And this is the way we do research. This is the way that academic centers like UNR and Renown are collaborating to be in the clinic, seeing what's happened, and to be at the bench and make new discoveries. And that's the exciting way that we've really developed all our drugs in cancer and in general for all diseases. Next, please. So you can't read this, but I want to just, what this slide shows you is that the first thing is done is to identify a potential target for cancer. Now, chemotherapy, in a sense, is targeted, but it's very broadly active, and the target doesn't matter as much. The problem with chemotherapy, traditional, what we call cytotoxic chemotherapy, which was developed now 70, 80 years ago, is the fact that it doesn't have a high therapeutic index, meaning it affects normal cells, as you well know. I'm sure many of you have seen the effects in your families or friends of chemotherapy. You've certainly seen it on TV or in the movies in an exaggerated fashion. But we know that you have to understand the biology. It's crucial to developing therapies that are going to help people. The important point I want to make is from the discovery in the lab to the time it becomes a drug that you can use, it often takes at least 10 to 20 years. That cycle is reducing, but there's a certain amount of time that's irreducible. Perhaps the fastest drugs in cancer, at least, that have been developed were developed in 3 to 5 years from the discovery to coming into people and to being shown to be safe and effective, and you can treat with that. But it's generally a little longer. Now, the good thing about validating a target, and these are just different examples of targets that were found 20 years ago or 10 years ago, and now we have drugs, is that once you have the first version of the drug, you can make the new and improved version. And almost all the drugs that are targeted therapies, we now are moving into the second generation, in some cases the fifth generation of drugs that work better, have less toxicities, because we learn how to tweak those drugs and work better against the target. So if you think about it as shooting an arrow, you're hitting the target in the first generation, but you want a bullseye, and that's what we're always trying to get to. Next. So there are 4 phases of clinical trials. This is in people. So after the preclinical work is done and a drug is actually developed that's felt to be able to be tolerated in human beings, that goes through these 4 phases. Phase 1 is where we look at safety, but phase 1 is changing because the drugs that are being developed today almost always are based on solid principles, and even if it's a new agent, a completely new class of drugs, by the time it starts phase 1, we can actually see hints of efficacy, shrinking tumors, for example. But we do a smaller number of patients to find the right dosage for different drugs. And then it moves into phase 2, where we test it in a group of relatively homogeneous population, like in one type of cancer and in one condition and at one point in their journey. And we see if that efficacy is really seen, and we get an estimation of what the benefit is. And then finally it goes into phase 3 studies where The study is designed to be very rigorous, a large trial, usually hundreds of patients, not thousands anymore because we've gotten better, but usually several hundred patients, usually randomized against the best standard therapy. In cancer, in fact, in general, we never use placebos if there's an effective drug that's out there. So we compare to the standard therapy. In rare cases where there's no active therapy, in other words, a patient's been through multiple types of chemotherapy, and the consensus is there really isn't anything that's been proven to help them, you might have a drug that just is tested by itself. But if there's a standard therapy, which today there is for almost every cancer for multiple lines, that's what it's tested against. So you might add something, or you might take away one drug and substitute a different drug. And these are usually done in what are called double-blind studies, which means that neither the investigator nor the patient know which they're getting a drug or a placebo. But that's in addition to the standard therapy, not instead of the standard therapy. If an effect is shown, then the FDA makes its decision based on two criteria, efficacy and safety. It does not think about cost. It does not really evaluate whether it's the fifth drug in the market or the first of a new class. It's really based on what the study shows in terms of efficacy and safety. And then once a drug is approved, we do continue to study it. That's what's called real world. And this is becoming more and more important because clinical trials in the past have been designed to be very specific. You have to meet multiple what are called inclusion criteria and exclusion criteria. You can't necessarily go in if your blood pressure is 150 over 90, even to a cancer trial if it's not controlled. You certainly can't go in if your diabetes isn't controlled or if your kidneys aren't functioning right. Well, in the real world, we know that lots of people who have cancer do have a degree of renal insufficiency and do have high uncontrolled diabetes. So we test these drugs in the non-clinical trial setting, but in the real world setting. And with our use of electronic data today, with electronic medical records, we can get very good data sets which really tell us what's going on in the phase 4 situation. Next, please. So in the last 20 years, those pillars have changed in emphasis, and they were really focused on precision oncology and immunotherapy. Those are the two areas that we've been focusing on. I'm going to spend a little time talking about those more generally. Next, please. So what is precision oncology? It's personalized treatment, and that is giving the patient the right drug at the right time and the right patient. The success of personalized medicine is knowing if a target is present or not. So just like you heard about lipoprotein A, you might want to test for something in a cancer patient, and there are multiple ways and multiple things to test for. And that way, if you know a target is there in a cancer, not sometimes what you're born with, but usually not, then you can use that drug, and you can spare people the toxicity if they're not going to respond to the drug. So therefore, we should have better responses and less toxicity. And that's what we call precision medicine. Next, please. Now, this has been going on for the last few decades. I'm just showing you the first targeted drugs, which occurred in the late 90s, and I was privileged to be a part of the studies that actually got both those first two drugs, rituximab, which is now a broadly used drug, not just in cancer, but in other situations, in immunotherapy and arthritis and so forth, which targets a type of cell called the B cell, and first the malignant B cells, which is lymphoma, and sometimes even normal B cells, because it does work there too. And trastuzumab, which was a revolutionary drug for a subgroup of breast cancer, which is what I mostly focus my career on clinically. And trastuzumab was the first drug against HER2-positive breast cancer. We now have multiple other drugs that work even considerably better. And the poster child is imatinib for a rare cancer called chronic myelogenous leukemia. That was a cancer that when you first got it, usually people were asymptomatic. Sometimes they had a large spleen, but they were usually found on a blood count. The problem was, at about 4 years in, it turns to acute leukemia, which is very resistant to treatment. We had no good therapies for this. We had only therapies to control the blood count, not to change the natural history of the disease. And I remember in my early part of my career in the 90s, treating patients with this disease and just helplessly watching as it changed. And we gave them strong doses of chemo, which helped very, very little when they became acute leukemic. In 2001, imatinib, or also called Gleevec, was approved, and this was a game changer. In my previous practice, I've been here 2 years. I spent 35 years in Memphis, Tennessee at the West Cancer Center and Research Institute. And when I left 2 years ago, every patient of mine with CML was still alive, was still on a pill. Most of them were still on Gleevec, although there's now 5 generations of CML pills. A couple of them had to go and change the pill. So that's the power of precision medicine. Now, unfortunately, not every cancer is like CML. CML has that one genetic problem. It's what we call kind of a dumb cancer in that sense. It doesn't develop additional genetic abnormalities. Most cancers do. And they're very, very clever at finding a way around, a path of resistance. And today, however, we have, this slide's from almost 10 years ago. At that time, in 2016, there were over 100 targeted therapies. Next slide, please. The targets are usually genetic. So we talk about born with genetic, as you heard about, lipoprotein A. You can be born with a gene like BRCA1 or 2, which increases your chance of breast and ovarian cancer substantially above the population. And because it has a role in DNA repair, which is critical for all our cells. But cancer is a genetic disease, by and large. So whatever happens, whether you're exposed to UV light to get a skin cancer, or alcohol to get a liver cancer, or whatever the situation is, in most cases we don't know what the cause is, or just age, and we're not repairing our cells. Every single cell that divides in our body has to replicate 3 billion bases every single minute. You can imagine that the body has a lot of mechanisms to make sure we don't get errors in the reading, but sometimes they fail. And when they fail, you get cancer. And we now have a whole bunch of what are called companion diagnostic tests that we can interrogate cancers for which mutations they have. The first genome was sequenced by the United States government, big science, cost $1 billion in 2001. It was an unbelievable achievement. Today, you can go get your full genome sequence for under $1,000. And there are companies out there that will do that for you. Problem is, we don't know what to do with most of that information, but you can have all the letters spelled out for you if you like at this time. But for cancer, we know a lot about it. Switching gears to immunotherapy, this has been the holy grail for, as I said, for decades, centuries even. In 1890, Dr. William Coley, who was a surgeon at the New York Cancer Hospital, which became Sloan Kettering, where I trained, were injecting patients with facial tumors with ground-up bacteria toxins. And you think it's amazing, but you know, we always look back on the previous year and say, why'd they do that? But he, if it didn't kill the patient, which it usually didn't, but it did give them very high fevers and felt bad, but the toxin in many cases shrunk the cancer. In some cases, it cured the cancer. This is in 1890, but we didn't really understand what that toxin, Coley's toxin was called, was doing. It was stimulating the immune system. It was a very, very non-specific way of doing it, and a very toxic way, but it was the germ, the idea that we can control our immune systems. And in fact, we know that cancer and our immune system, when you develop a cancer, you are locked in an eternal battle. Most times the body wins, the immune system wins. And we think, the theory is, that many of us are developing cancers that are subclinical, and as they develop, our immune system takes care of them. But as I said, cancers are tricky, and they have evolved to develop mechanisms to shut off the immune system. So the group of drugs that we're using most commonly for immunotherapy today is called immune checkpoint inhibitors. We don't want our immune system to run wild, right? It's a very delicate balance. It should work when we have an invader like a bacteria or a virus, but if it keeps working, and we've seen this in the context of COVID too, you can have problems with that, with autoimmune or long COVID and the symptoms there that we don't understand very well. So our immune systems have developed evolutionarily over millions of years to be very fine-tuned with a lot of checkpoints. And cancer usurps those checkpoints and turns them off. What these drugs do is take the brake off. So cancer puts the brake on the immune system, and these drugs take the brake off. And they're antibodies for the most part. Can you go back for 1 second? I'm just going longer because I guess we have time. So there's a lot of other things in cancer, and you mentioned mRNA. So mRNA vaccines, using the technology of COVID vaccines, are in the clinic now. They're in clinical research. So far, we've seen very encouraging early results with a type of cancer called melanoma, which tends to be one of the most sensitive to immune stimulation. And so we'll see. I would predict that we'll have mRNA vaccines against cancer actually available that are approved within the next 5 years for certain cancers. And then there are many other ways to do it, including things like cells. We can now manipulate cells. The immune system is part antibodies and part cells. The cells are actually, in many ways, the most important part against cancer. So we have cells called T cells, which go around in our body like Pac-Man, and if they find a bacteria, they're going to chew it up, or they chew up a cancer cell, and it's stimulated. So we can create cells now called CAR T cells, and that therapy is being used mostly against lymphoma and other hematologic malignancies. It does have a lot of toxicity. It's done right now in specialized centers that do bone marrow transplant. We hope to bring that to Reno and Carson in the next 5 years as well. Next. So as I said, the immune checkpoints have been a transformative new pillar of therapy. The first drug was approved 10 years ago. We participated in those phase 3 trials in melanoma that were really striking. Today we have multiple drugs, as you see there, with different checkpoints. And there are over 3,000 clinical trials going on with checkpoint inhibitors exclusively right now. That's the power and the belief that these will transform things. Next, please. These immune checkpoint inhibitors work broadly. I don't think you can read that, but almost every cancer in almost every organ will have some success with checkpoint inhibitors. In some cases, they may be curative. We're not far enough out yet, but in melanoma, the skin cancer, which was deadly before if it had spread, there are patients now who have been treated with these drugs that are in complete remission and off therapy for 10 years or more now. People that had cancer throughout their entire body in remission and potentially cured. The survival rate before those, when we had just traditional chemotherapy, which doesn't work well in melanoma, 6 months for a patient like that. So really profound difference. Next. And I just want to point out one other disease, lung cancer. So I mentioned lung cancer at the beginning as the biggest killer and of men and women, and it's still a problem. Even though we've been very successful at reducing smoking, only about 15% of adults today smoke, and that's good, but we still have work to do, especially preventing adolescents and young adults from starting to smoke. But lung cancer is still a problem. When I started my career in the late 80s, yeah, I'm really old, but we debated, even into the late 90s, should we even give chemotherapy to patients with advanced lung cancer? Are they really getting any benefit, or are we just torturing them with that for a little bit of shrinkage of the tumor? And we did make some progress there, but it was very modest. Look at this slide. Each of those circles represents a new drug approved since 2003. Those drugs are not chemo. Those drugs are personalized medicine, Turns out lung cancer, which I would never have predicted 20 years ago, is 50% of lung cancer has a single mutation driver, different ones, about 8 or 10 different ones. There's a drug for all of those. If you have, and we test for all of those. You get lung cancer today, you should have what's called a comprehensive genomic profile at any stage of lung cancer. That's the new standard, just emerging, but has been the standard now for a few years in advanced lung cancer. And the other drugs are immunotherapy. So immunotherapy worked very well in lung cancer in combination with chemotherapy. And we now have patients living 5, 10 years with advanced lung cancer, with disease stable or even in remission. Not ready to call that cure yet, unlike melanoma, but it's definitely a huge advance. And for these patients, they get to live their lives longer. They get to see their grandchildren born. They get to go to weddings. It's really meaningful. Next, please. Same thing is true in blood cancer. I won't belabor the point. We have a lot of new drugs, and cellular therapy, as I said, is true as well. Next, please. Last thing I want to say is that medical research, as Kristen said, is a team sport. You're a part of the team, and being here tonight is very meaningful and gratifying to me. You're interested in clinical research, and hopefully you'll be ambassadors for clinical research with your friends and your families. There's still a mystique that people are guinea pigs. They're not guinea pigs. Every patient, I'll reiterate what Dr. Block said, every patient who's getting a therapy today for any disease is because other patients put themselves out there to do a clinical trial, and they would not know if they would benefit or not. They wouldn't know if they were in the group that would benefit or not. But they did that because future generations, possibly them, but future generations benefit. And the same is true today. The only way we make progress is for people to volunteer for clinical trials that are well designed, and that we're trying to do. So besides the patients and their families, who are most important, interested people, it's everybody else, the healthcare systems, having a wonderful clinical research office like we do at UNR and Renown, and that makes all the difference in the world. Clinical research is unduly complicated. I could go on for hours. I mean, you would not believe the amount of regulations that that control clinical research. Now, that is good. We know about a lot of bad things that were done in research in times before these structures were developed. But we now are in an era where we're kind of saying, how do we make this simpler? How do we reach more people? Do we really need all these inclusion criteria and exclusion criteria? Because that's not real people. And it takes longer, and it's more expensive. And how do we get medicines to people? How do we get medicines to diverse populations, which we, frankly, have done a poor job at? If you look at the African American population in the country versus the African American population in clinical trials, it's about 6-fold higher. So we don't know a lot about the drugs that get approved for everybody. The FDA has taken a very strong stance on that to make sure that there's diversity in all clinical trials going forward, and pharma has done it as well. We have to depend on philanthropy. We have to depend on citizen advocates like yourselves, the regulators. FDA has a tough job. They don't want to be too lenient or too restrictive. They are good professionals. I've gone with many pharmaceutical companies to the FDA to design trials. They're there to help. They have a lot of expertise. They do have a lot of regulations too, and sometimes you can massage things as when you sit there and talk with them. But they're really on the side of the public. Next slide, please. So finally, for our Pennington Cancer Institute at Renown, we have now about 30 open trials with pharmaceutical companies, as well as National Cancer Institute sponsored trials, so-called cooperative group trials. Right now we're focusing on breast cancer, colorectal cancer, esophageal cancer, and other GI cancers, and lung cancer. As we build our program, we just recruited a lymphoma expert who will be doing trials in hematologic malignancies starting next year. We're very interested in immunotherapy and precision medicine approaches. I told you about the immunotherapy drugs, the checkpoint inhibitors. We're combining them now with the concept that getting 2 checkpoints open might be better. There's a couple of drugs already approved. A particular driver cancer gene might be present in only one cancer, but usually they're present in multiple. So we call that tumor agnostic. And if you have one of those, we can use the same drug in multiple cancers. We're looking at that. We're looking at finding detection of cancer in liquid biopsies. One tube of blood now you can do to find, I would call it a needle in 1,000 haystacks, DNA from the tumor that is shed, even when there's a small amount of tumor. And we can test repetitively to see, after you give treatment, if that mutation goes away or down or goes up, and it heralds relapse or progression. So that's a very fascinating area that we're doing. The NCI groups I mentioned, we're doing from phase 1 through phase 4 now at Renown, and we're hoping to expand our phase 1 capabilities in the future. And it's all in conjunction with our wonderful clinical research office at the University of Nevada, Reno. So that's it. Thank you for your attention. I'm happy to take some questions.
Thank you, Dr. Schwarzberg. That was an excellent overview of cancer therapy, and cancer research. You know, the word cancer evokes a lot of feelings and emotions in people. And I'm sure everyone here has been touched by cancer in some way, either personally, family member, or a good friend. So thank you very much.
Thank you.
So I have a few questions here. Well, I don't know if you'll be able to answer this or not, but if you receive a questionable or abnormal test result, will insurance pay for a second or third opinion? And how do you go about getting that opinion from a highly rated specialist or research hospital?
Okay, yeah, that's that's a tough question. In general, most insurances actually do pay for second opinions because when you think about it, they want you to get the best therapy, which usually turns out to be the most cost-effective as well. But it does depend on insurances, and so you really have to check the fine print. But I would advocate for second opinions. I've always found that doctors in general want their patients to get the best, and they're more than happy to recommend, and I am too, if a patient asks for a second opinion. And so it should not be a problem. We have a number of academic institutions in the area where second opinions can be had, and they work with us closely. Of course, our overall goal is to bring all that academic expertise right here at home, so you don't have to leave our communities to get the best care.
So this next question is similar, and I hope you have a good answer for this.
Okay, you put the pressure on.
Does your family doctor or primary care doctor, are they required to refer to a particular doctor for a second opinion, A, and then B, how does a patient select the most appropriate physician for their second opinion or their?
It's a really good question that I'm going to say there isn't a good answer to, in the sense that, you know, you can look up Consumer Reports or you know, just Google anything and get a lot of data around not outcomes, but characteristics about a product or things. You know, there's a lot of reviews. We have that available. That doesn't really exist in medicine. So there is, to some extent, a black hole, I think, in terms of how you get to the care you need. I would hope that, first of all, I will dispel the concept that people are told who to refer to. I mean, I think almost all physicians have your best well-being at heart, and they're going to refer to who they think the best. But again, it's hard for them to parse that out as well. I do think clinical trials is frequently a separator. It's usually an imprimatur of high quality because companies only work with people that can do rigorous studies and know the field well. So people do clinical trials in general are experts in that area and sometimes in that disease. So that's one way to look at things. Obviously academic centers, but there are many, many fine community-based oncologists. I can only really speak for oncology here, but they exist in every state, in every country. The training programs are excellent, and all over you can find good. And so, but it's not an easy task. I will grant that.
So should a patient rely on Yelp, Facebook, Twitter to choose their doctor?
That one's a little easier. I would say no. Sometimes the people that are most publicizing themselves are doing it for reasons other than their expertise. I'll just leave it like that. But I do want to say one thing about internet and cancer, and this is something I've seen evolve over 35 years. Of course, pre-internet, there were other ways of doing it. It's just much easier now to take advantage of vulnerable people, and it really makes me angry that people are on the internet. You're clinging for hope, right? But you have to have realistic hope, and your doctor should be straight with you, be honest, but be there as a partner, and not, we have the cure for cancer and 16 other diseases with my therapy. Send 1995 to get the first dose. Don't do that. But a lot of people do.
So, Kristen, you haven't been forgotten. This question is for you. Is Renown doing any clinical research in regards to dementia, Alzheimer's, Parkinson's?
Ooh, that's a great question. So we do have an ongoing Parkinson's study, and we did have an Alzheimer's trial that has recently closed, so we're looking for a replacement study so we can provide that care. But we do have neurologists who are doing clinical trials. We're also branching into the multiple sclerosis space. So if you have an interest in that, definitely reach out, and I'd be happy to chat with you about that portfolio.
So are there any other,
And
I think our third speaker.
she's here.
She's here.
Yep.
Where is she?
Oh, she's here. I went she wasn't.
If you want to stand up and just stretch for a second. Thank you. So Dr. Farah Lovely has over 20 years of experience in practicing at Renown Health in pulmonary and critical care, which means she knows more about medicine than anybody else here tonight. Currently, she's the Assistant Dean of Clinical Research at the University of Nevada, Reno School of Medicine. Dr. Lovely.
Thank you, everyone, for being here. And I'm sorry I was late. I was actually working in the ICU, and my backup came late. And this was a little far from South Meadows, so I'm sorry about that. All three of us different speakers have taken a different approach in presenting to you all. So hopefully we'll give you the information you want and are interested in. So I can go ahead to the next slide. And I kind of put it as clinical trials in Northern Nevada because I wanted to walk you through the journey of how we've taken this path, this steep path uphill in expanding clinical trials here. So next slide, please. So our research journey first began with COPD. How many of you know what COPD is? Okay. So there's now valves that you can put in emphysema areas that can make the areas smaller of emphysema and allow the normal part of the lung to expand more. And it's shown to improve life by 1,000 days, which is, for people with end-stage emphysema, quite a bit, and improve quality of life. So part of that has also been when somebody has a collapsed lung, there's humanitarian clinical trial where you can put these valves, and particularly in emphysema patients, when their lung collapses and doesn't expand all the way, and this is part of a clinical trial. And that's kind of how our journey started with research. And of course, we have internal medicine residents, so we have case reports, because this is their journey in publishing and their interest in research as they pursue specialties or non-specialties, but as they pursue their career goals. And then came our pandemic. We have not forgotten, have we? So how we realized in Northern Nevada when we wanted testing kits, guess where all our kits went to? What's that street? What's the street with tons of lights? What's that city called? Las Vegas. Everything went to Las Vegas. Absolutely everything. And we weren't getting anything. They were like, well, if you're part of a trial, you can get some. And we just realized, even all, like, remember the plasma therapy, remember all these medicines you were hearing about? Well, we realized we were nowhere. I mean, Kristen and I were working together at that time, and we started signing up for clinical trials so our patients could get access to a lot of these medicines. So that's kind of how our journey started, where everybody was like, what are you doing? What are you doing? We're like, we're trying to figure this out ourselves. Hold on. But, and we even had a provider fly his own plane to go get us plasma, as part of this trial for somebody. It was amazing. But everybody got together, the team got together, and that's kind of how our journey started into this really uphill course that has really progressed into something wonderful. And providers like Dr. Schwartzberg are here, bringing much more to Northern Nevada that we didn't have before. So this is absolutely great. Next slide. So, Dr. Schwartzberg talked to you about clinical trials, phases. I kind of wanted to take a step back in terms of clinical trials, also observation studies, not to forget about what that means. During our pandemic, remember hospital beds, we didn't have enough of them. So many patients with COVID would go home and we would monitor them from home. It was called remote patient monitoring. And we also used it for COPD patients because exposing, in the hospital, that was like the worst place to be. So we didn't want our patients with COPD to be in the hospital, so we monitored them from home with much higher oxygen needs. And those are the kind of studies we are after, we weren't doing it for a study, but then we had enough data to look at it on a study and see, patients got to be home with their families, they were being monitored, we had a nurse calling them to check on them. And we saw that with our patients with both COVID and COPD, that people, and it's an observational study, that it decreased the hospital length of stay almost by 2 days. Can you imagine the cost of the overall cost of healthcare, especially when we run out of beds soon, as everybody's moving to Northern Nevada because it's a very well-kept secret, and we're all discovering how beautiful it is. But we don't have enough hospital beds. We just don't. And so, this is our future, but this is kind of observational studies. I gave an example of knee injuries. Are you at risk of osteoarthritis when you have knee injuries for all you skiers, It is a risk, and that's kind of part of clinical trials is observational studies. Okay, next slide. So we talked about lung cancer, and put this slide up because screening of lung cancer, look at that statement. In Nevada, 1% of those at high risk were screened, which is significantly lower than the national rate of 6%. So the national rate is horrible, but our rate is horrible. And we're at the bottom of the list. California is below us. So hey, we got one step up. So next slide, please. So that's one of the clinical trials we're doing is in genomics. We're looking at mRNA, not the same as the COVID vaccine, but mRNA, nasal swabs. We're working with a company where we can do a nasal swab. So when there's a cancer in the lungs and the mutation is there, it's supposed to be in the entire airway. So when we nasal swab you, just like that COVID test, that's where we see whether you're a low risk, moderate risk, or high risk. And we finished the familiarization study with really good outcomes. It was over 90% correct, per se, in that study. And now we're going for the little bit larger nodules, spots in the lungs, and they're 8 millimeters to 3 centimeters. So you have a CAT scan for whether you do lung cancer screening or incidental, you just so happen to get a CAT scan because you landed up in the ER or urgent care and got a CAT scan done. A spot is seen. If it measures that, we contact you if we see that CAT scan and say if you're interested in the study. So that's one of the studies we're doing, and that's the best part. Before we used to have to do a bronchoscopy where we'd put a camera down into your lung and then swab the inside of your airway. Now it's just the nose, which is great. I guess none of it's quite great, but so next slide, please. And this is the Nightingale study, and we've presented at multiple centers, and we're working with about 35 sites. Yeah, with about 35 sites, we're working together and hopefully by next year, this will be the standard of care. Next slide. Chronic lung disease. So, this, of course, I'm a pulmonologist as well, so lung disease is my forte. But how many of you have, when you've had a cold or viral illness, or even if you have lung disease, that shortness of breath, that how you cannot breathe, even if you have like an asthma attack, like where you just can't take that next breath, and you can't, and you're leaning over, you're leaning like this, trying to get that breath, it feels awful. You're exhausted all day, you can't function, and life just can't, you can't seem to catch a break because you just don't feel good. So when you take a deep breath, what your diaphragm, which is the muscle that separates your lungs from the rest of you, you take a deep breath, your diaphragm should go down. You relax, your diaphragm should go up. So that's one of the studies we're looking at. So when you have chronic lung disease, one of the things we found, and that's over decades of studying lung pulmonary rehab, where we teach you how to breathe again and how to take breaks and how to improve your muscle strength so that you can go grocery shopping and pick up a grocery bag, walk up a flight of stairs, even if you have chronic lung disease. But we don't know how to objectively measure it other than have you walk for 6 minutes back and forth and measure that distance. So one of the studies we're doing, and so that's next slide. Let's go to the next slide. I kind of just jumped right into it, is looking at dynamic digital radiography. So when, you remember I said when you take a breath in, your diaphragm goes down, you take a breath out, goes up. So we're looking at that distance after 9 weeks of exercise, when we teach you how to breathe again and teach you how to use your muscles correctly, build these muscles up, build these quadriceps up in your arm muscles so you can pick up grocery bags, go up a flight of stairs without getting short of breath. We want to see if we can improve that muscle strength. So with this type of x-ray, which you can see the diaphragm go up and down, we're wanting to see, can that distance increase? Medicare only pays for 9 weeks of this exercise. And we know for a fact, after 26 weeks, your muscle memory is gone from that diaphragm. We know that. So if we can show how much we improve it, we can probably show, maybe, is the theory, later on, that, hey, the diaphragm muscle excursion decreased, so now they have to get another 9 weeks of exercise. And so that's one of the studies we're doing. And we're also using an ultrasound to actually measure the diaphragm. And this is sponsored by a Japanese company, which is great. And we have about close to 20 patients enrolled in our study right now, and we're hoping to hit 100, which we think we will by end of the year. Next slide. And that just keeps, this is 99 patients actually is our target, not quite 100. Next slide. Viral assays. So I talked about our journey with the pandemic, about looking for just the tests. So now we're partnering with different groups to have different tests. So how fast, how accurate, and I don't know how many of you had those COVID tests, but remember when it was like a brain biopsy where they shoved it so, I don't know if any of you went through the CVS where it was just awful. So now they're testing where you don't have to get your brain biopsy done to get a COVID test. It's just on, so those are the kind of tests we're doing. And this is one of the companies we're working. Actually, Kristen and the team are coming into the lung clinic to offer that test next week, just in case anyone's interested. Next slide, please. Next slide. And then pulmonary fibrosis. How many of you know what that is? Yeah, it's lung scarring. And it's more prevalent. We're seeing it more and more in ages 65 and older. Some of it's genetics, and a lot has to do with our environment. So genetics, our smoking history, and our environment. Like miners, uranium miners. Granted, we don't have uranium miners here. We have them in New Mexico, but uranium miners. That's a classic scarring of the lungs that you can get. But we're seeing many, and we call it idiopathic, unknown cause, because we know a lot of chemicals cause scarring of the lung, but we can't say you have it because of X, Y, and Z. We can't say that if it's not one of the ones well studied. But how do we slow it down? We can't. It's a progressive disease. There are 2 drugs in the market, and they're called antifibrotics. They're $10,000 a month. There are only 2 FDA-approved drugs. So that's one of the trials we're doing. It's another drug, which is an anti-inflammatory drug that we're hoping whether it's in combination or not in combination with the idiopathic pulmonary unknown cause scarring of the lung, or progressive fibrosis, so people with lupus or other diseases that have scarring of the lung, or known cause, like exposure, something called bird fancier's lung, where they're exposed to bird feathers and develop scarring of the lung because they have their immune system reacted to it and scarred up their lungs. This is a drug we're studying currently. We're in a clinical trial with multiple people around the world, multiple hospitals, clinics around the world. And this is one of the ones we're studying to see. And this is one of those where Dr. Schwartzberg was describing a phase 4 trial. So if everything goes well over the 12 months, this would be a trial. Patients can stay on the medicine continually by the company as we wait for FDA approval. So this is something we're testing. Next slide. And how when you cough, right? This is one of the trials we're doing. So cough has multiple reflexes, whether it's nerves, a trigger from the diaphragm, all the way up from the brain, all the way down to that diaphragm, and even below the diaphragm can trigger your cough. There are many sensors, but when you cough, how do you know? Is it a mild cough? I heard a cough, but I don't know how to describe. Is that mild? Is that moderate? Is that severe? If you came to see me in clinic today and said, oh, I cough all night, I can't sleep, I'm miserable. To you, that's severe. But how do I know when I give you something and treat you that it became moderate when you still can't sleep all night and you're blaming the cough? Maybe it's something else, but there's no way for us to measure it. So there's a company that has a device. So that's what we're testing. And we're working with our medical students and our patients and who enroll in the trial, and it's via telemedicine. They're given a gadget to measure the intensity of the cough. So can we tell from the volume and the frequency, is this a mild cough? Is this a moderate cough? Is this a severe cough? Or where does it fall on the spectrum? And then that helps with developing medications, developing and assessing if you're improving, not improving, and also psychologically can help people as they deal with their chronic cough. Next slide. And then, of course, the age-old COPD. How many of you smoked in high school? You guys have to all put up your hand. Did nobody smoke in high school? Really? Carson City did not smoke in high school. Wow. How many of you had parents that smoked? Okay, now we're getting somewhere. Okay. So, I mean, women are more risk for COPD than male. Secondhand smoke can result in COPD. And exposure to your environment can also result in COPD. But COPD is a pretty prevalent disease in our nation. And it's pretty prevalent here, especially as when the people who've had to work in the casinos when smoking was so prevalent. So people who've spent their careers working in casinos have developed COPD. Has anyone paid for inhalers lately? They cost an arm and a leg, right? And Medicare keeps changing their mind who they're going to sign up for with which inhaler. So sometimes it's $700, sometimes it's $30. Yeah, no, these are facts. And the hospitalization, because when those we lucked out this year with no fires. We definitely lucked out. I mean, we all complained about the snow, but thank you for the snow. But when those fires come, you think those window seals that we all have, they're not completely sealed. I mean, smoke is coming through. And the particulate matter, like the little particles, are all coming through. No matter how many filters we all buy from Sam's Club or Costco, it's not enough. And many COPDers land up in the hospital. So the question is, how do we prevent that pathway, just like in asthmatics, that trigger of the pathway of the inflammation that just gets escalated to not land you up in the hospital and not land you needing steroids all the time. Prednisone, the steroids, that results in bruises, broken bones, high blood pressure, diabetes. Exactly. That's what we're looking for. That's what we're trying to figure out. What else can we use? Because the inhalers only help with symptoms. There's no cure there. There's no blockage. It doesn't prevent the exacerbation, the attacks of COPD. So what we're testing is an injection that blocks the pathway that triggers the inflammation for a full-blown attack. So you don't need steroids. You don't land up at the hospital. And they've done this with asthma since 2013. There's now 11 of them on the market. Asthma is great. There's so many options with asthma. But COPD only has these $700 inhalers or $30 inhalers, and it doesn't protect from the inflammation. So that's what we're testing, and it's a 36-month study. And it's the next couple of slides. Yeah, and this is our age group, 40 to 80. You have to have had 2 attacks or needing steroids and or antibiotics twice in a 12-month period. And this is a worldwide study that we're doing as well. Okay. So other studies, we work with our medical students very closely. So World Lung Day, September 25th, everybody, next Monday, just so you all know. Do you think there's vaping in high schools here? Yes. Thank you. Okay. No smoking in Carson City, but there's vaping. Okay, so yeah. Yeah, and have you seen the number of chemicals in vapes? And it's permitted, right? Everyone can vape away. So we're doing an anti-vaping campaign at some high schools. So the medical students have built models of lungs and the destruction of lungs, and then built a game with chemicals and what damage it can do to the person next to you as well as to you. And then they do a test for the high schoolers. And the WashU school, we started small because we're trying to figure out if this is a good study to do. And it's a survey for the medical students, how confident they are pre and post teaching in a public event. And that's the key, right? Part of the research is not only to be for us to be an advocate for our next generation, but also to bring teachers into the next generation. So this generation of med students, to help them participate in a research study so they can be teachers and help the next generation. So this is that's kind of one of the projects we're doing. Other projects, health literacy projects, we're working with pediatrics, so understanding how do parents understand their child's milestone in writing and reading. So helping parents understand that, and how do pediatricians educate the parents. So that's one of the surveys we're doing. We're also working what we call domestic global health, and that is our immigrant population, or undocumented, as well as our undocumented immigrants, as well as our refugees, to help us understand what they don't understand, and then help educate them in chronic health disease. And that's one of the other studies we're doing with our School of Medicine. And I think that was my last slide, other than questions. But I forgot to mention one other study. We do have a hypertension study as well. Why, as a lung doctor, I'm doing hypertension, I'm not sure. But no, I'm also internal medicine, so all good. But we're testing a drug down the pathway of aldactone, which triggers the renin system that increases blood pressure. So anyone on 2 or 3 medicines for blood pressure, we're trying to see if we can substitute for a combination medication. And that's a study with a group called Mineralis, and that is also worldwide that we're doing. So that's the only one I didn't mention here. But now I'll take questions.
Well, thank you very much. That was a great overview of what's happening with pulmonary research in medicine in Northern Nevada. I guess my first question is, when do you find free time?
Hence, I was late.
So any questions from the audience? Or?
So currently, the inclusion will be your lung function test has been decreasing by at least 10%. The idiopathic one did close because they maxed out their number of patients, but the progressive one is still open.
Treatments, and I'm wondering what your thoughts are on stem cell
So the stem cell and pulmonary fibrosis, and actually they're using stem cells, so some of these stem cell companies are doing it for many chronic diseases. They haven't published enough objective data to show us clinicians. So when we look at a study and we're looking, how Dr. Schwartzberg said, how safe is it and how efficacious is it? Like, did your lung function change from 1.6 to 1.8? That's the absolute change. Or can you walk more?
On a scale of 1 being dead and 100 being perfect lungs, the first test that I got, I was at measurement 42, and I went up 5 points about 6 weeks later, but then it started trailing off again, and the second time I only went up 3 points.
Yeah, and
Now I'm well under 40 now, and
but that's why with stem cells, they haven't been able to prove the objective, the absolute objective. And the points, the way we look at points in terms of not percentages, but by milliliters. So 200 milliliters is an absolute improvement of your lung function if you increase by 200 milliliters, or if you can walk 34 meters more than you did the last time, or your oxygen requirement went down. So those are kind of where we look for efficacy of the drug, like did it make these objective improvements? And there's a bunch of others, but those are what we look for. And with stem cells, it's not ready for prime time. They've not been able to show us that.
they're very expensive.
It's yeah, that's, I know, yeah.
Any more questions for Dr. Lovely or a panel? Well, I see a question coming, but as far as our panel is concerned, do any of you have any final comments or anything you'd like to add that you didn't cover or want to cover? Or?
No, we just want to thank you again for coming out and your interest, and just keep us in mind because, as you heard, we're a very active and growing program, and we're here to serve the public.
And I will add too, I do have business cards, so if you didn't catch our contact information up there, I'm happy to pass that out to you and you can reach out to me directly. Thank you.
So I'd like to thank the panel, Kristen Gurney, Dr. Schwartzberg, Dr. Lovely, for taking your time out in your busy schedules to be here tonight. Very enlightening and very educational. I'd also like to thank the Sierra Nevada Forum for organizing this. You guys always do a great job. Thanks for everyone for coming. Thank you.